Good afternoon, everyone. It's a pleasure to attend this webinar on gastroenterology and hepatology. I would like to thank the organizers, of course, for the opportunity to disseminate the results that I achieved during my PhD at the University of Bologna. In details, we observed that fractions obtained from the leaves of Arbutus unedo exhibit chemotherapeutic properties for the treatment of gastrointestinal stromal tumors. Gastrointestinal stromal tumors, typically referred to as GIST, are soft tissue sarcoma that originate due to oncogenesis in mesenchymal stem cells that should differentiate in interstitial cells of Cajal, cells that are crucial for the regulation of gastrointestinal motility. No treatment options were possible for patients with unresectable or metastatic GIST due to unsatisfactory results observed in clinical trials, as you can see by the low partial response using regimens based on traditional chemotherapeutics such as doxorubicin or paclitaxel or many others. The scenario completely changed when it was discovered that most of GIST harbor, almost 90% of cases harbor gain-of-function mutation in two tyrosine kinase receptors, KIT or PDGFR receptor alpha. This mutation lead to the aberrant activity of the receptor, which is independent from the regulation promoted by the binding of their endogenous ligand, leading to the activation of numerous pro-oncogenic pathways. For this reason, the use of imatinib, an inhibitor of tyrosine kinases, including KIT or PDGFR receptor alpha, completely changed the perspectives of patients. As you can see in the picture, Gleevec, the brand name of imatinib, increased the median overall survival up to 57 months with respect to 10 months observed in a cohort treated with doxorubicin. Unfortunately, imatinib, as you can see in the treatment outcomes, promote complete response in just 5% of patients. The most of patients show partial response or stable disease instead, a condition that is only transient. Due to the growth of imatinib-resistant subclones that lead to imatinib-resistant and consequently to progressive disease within 24 months. Further mutations were commonly found in both tyrosine kinase receptors in imatinib-resistant GIST, in particular in the ATP binding pocket and the activation loop. Preclinical studies demonstrated that the presence of these mutations can counteract the mechanism of action of imatinib. For this reason, the research was focused on the identification of novel tyrosine kinase inhibitor to target this panel of mutation indicated in the slide with the red signal, leading to the development and the approval in series of sunitinib, regorafenib, and ripretinib, the last one obtained in 2020 with the best targeting profile. However, as you can see, the use in series of these tyrosine kinase inhibitor lead to a modest increase of median progression-free survival. In addition, ripretinib, as the best-in-class TKI developed, was proposed as better second-line treatment for imatinib-resistant GIST. Unfortunately, as you can see at the end of phase 3 trial, ripretinib was not superior to sunitinib in terms of PFS. This result is extremely important because confirm that a better targeting profile for secondary mutated KIT or PDGFR receptor alpha is not sufficient to have a better clinical results. This result is corroborated by numerous preclinical findings that have demonstrated that there is a plethora of mechanisms that could be present in yeast. For example, in the slide I have indicated axle upregulation, MET or microRNA, long-coding RNAs. There are different mechanisms that cannot be targeted by multi-target TKIs. So, the multifaceted landscape of imatinib resistance confirms that a strategy based only on TKIs is not cannot be the solution for targeting GIST. So, in this context, the multifaceted landscape of imatinib resistance cannot be targeted with a molecular targeting approach. Instead, the identification of a traditional chemotherapeutic that is defined as no specific cytotoxic drug could potentially allow the targeting of more imatinib-resistant subclones independently from their mechanisms of resistance. However, as I described at the beginning of my presentation, different chemotherapeutics were tested in GIST, but nobody of them showed efficacy for the treatment. So we decided to start a drug discovery project to identify novel potential chemotherapeutics for GIST treatment. In particular, we focused on plants, and in particular a crude extract obtained from the leaves of Arbutus unedo that was already associated with an anti-cancer activity in osteosarcoma cells. We hypothesized that in this extract could be potentially present novel chemotherapeutics useful for GIST treatment. Therefore, we first evaluated if the effect observed by Cappadone and colleagues could be also present in G cells. As you can see, the treatment with AUN reduced the viability of G cells in a dose-dependent manner. Noteworthy, the reduction in viability is associated with the rise of a population that is annexin 5 positive, a well-recognized marker of apoptosis. Since AUN also show effect in G cells, we decided to investigate which phytochemicals could be responsible of the observed activity, and we decided to apply a bioguided fractionation in order to divide phytochemicals based on physical and chemical properties, and to finally identify them using analytical techniques. In particular, we started with a liquid-liquid partition and divided phytochemicals contained in AUN in 3 different solvents: chloroform, ethyl acetate, and water. And then we tested this subfraction of AUN in G cells. As you can see, the FR2 is the most active fraction, and in particular in the apoptotic profile with annexin 5 labeling, as you can see, FR2 reproduced the effect that we initially observed with the UN, indicating that the phytochemicals, the active phytochemicals contained in the UN were divided, were present in FR2. Once identified FR2 as the most active fraction, we proceed with the second step of bioguided fractionation. In this case, we apply the reverse phase medium pressure liquid chromatography, obtaining 4 additional subfraction. As you can see, this was our workflow in which we observed the elution of phytochemicals using UV signals, applying elution through a gradient of solvent, in this case methanol. Then we evaluated the pharmaceutical effect, the pharmacological effect of the obtained subfraction, observing that FR2A represented the most active, showing a pharmacological effect in G cells in terms of viability reductions that was comparable of the effect that we observed using a higher concentration 200 micrograms ml of FR2. Since the results obtained with FR2A in terms of viability reduction were promising in G cells, we performed an experiment in order to compare the effect of FR2A with respect to the gold standard imatinib. And as you can see in this time course experiment, the treatment with the two different concentrations of FR2A, 33 and 66 micrograms ml, reduced the viability, whilst the treatment of imatinib is statistically different with respect to the control only in the last time point. This is an expected result since imatinib, as you can see, induce a faint increase of apoptotic cells. The main effect of imatinib in G cells is the prevention of cell proliferation and growth. Instead, the treatment of cells with the subfraction of AUN induce the rise of apoptotic population. We then tested the efficacy of FR2A in a broad panel of cell lines, in addition to GIST-882 already used in previously validations. As you can see, FR2A affect also the viability of GIST-1 with a similar IC50, and moreover, it can also target imatinib and in general TKI resistant cells such as GIS48 and GIS48B, even if with a slight higher IC50. In particular, the effect on GIS48B is particularly relevant because this cell line is KIT negative, and suggesting that the effect promoted by FR2A is independent from KIT expression. Interestingly, even if we observed an Exin5 positive staining, we didn't observe the traditional cleavage of caspase-3 or caspase-9, but we observed that FR2A reduced the level of PARP1, even if the traditional cleavage typically associated with the activation of caspase-3. This suggests that the mechanism of action of FR2A could be associated with the PARP1 deregulation in a caspase-independent mechanism. We then tried to identify which phytochemical could be responsible of the activity of FR2A. Using NMR, we identified beta-arbutin as the most representative molecules contained in FR2A, a molecule that was already reported to be the main constituent of AUN and was already associated with pro-apoptotic activity in melanoma cells. Unfortunately, these molecules, the pure beta-arbutin in GIS-82 didn't reduce the viability, indicating that it was not the active phytochemical. We decided to perform another step of back-added fractionation because the further molecules containing FR2A were not sufficiently concentrated to be identified with NMR. So we decided to enrich their concentration with a further back-added fractionation step. So we applied a size exclusion chromatography and a T-layer chromatography to divide FR2A in 84 different subfractions that were subsequently tested in G cell lines. In particular, we identified 9 subfractions that reduce the viability of G cells. In particular, we divided them in 4 clusters since we observed that active in series subfraction were present, suggesting that probably the same phytochemicals was present in this subfraction. And we also identified a cluster, the 2A3536, that was more potent. The take-home message of this experiment is that in FR2A there are at least 4 different active phytochemicals, since we observed a reduction of ability in 4 different clusters. Once obtained the subfraction of 2A35, we applied this workflow to identify, using NMR, which could be the active phytochemicals. In particular, we compared the spectrum of the active subfraction indicated in blue with respect the adjacent weakly or non-active subfraction. We applied this method to identify which signal were exclusively present in active subfraction or more concentrated in active subfraction. In this case, in the cluster of more potent subfraction, we identified gallic acid, myricetin hexosid, and galloyl arbutin as potential active phytochemicals. We applied this workflow to the 4 clusters that we identified, observing that active subfraction always contained pyrogallol-bearing compound that were all characterized by the presence of this moiety, the pyrogallol. And interestingly, these molecules and this moiety was always absent in the weakly or non-active subfraction, supporting the concept that pyrogallol-bearing compound were responsible of the effect that we observed in yeast cells. We then tested gallic acid, because gallic acid was present in the more potent subfraction, and we compared the effect of gallic acid with respect to 2A35, the subfraction that contained gallic acid. And as you can see, gallic acid is active in G cells, supporting that gallic acid could have a pharmaceutical effect in G cells. But interestingly, gallic acid is not active when used at lower concentration, indicating that in 2A35 could be a phytocomplex with more compound that are responsible of the activity of 2A35. So, since our aim was the identification of a novel potential chemotherapeutic, we tested the subfraction that we originated in PBMC, peripheral blood mononuclear cells, in order to see if this compound could have a non-specific effect and reduce the viability also of PBMC. As you can see, these clusters affect in a similar manner both GST1 and PBMC, an effect that is also observed using doxorubicin, an approved chemotherapeutic that is not effective in GIST. To conclude, we have applied back-added fractionation to the extract of AUN, and we have derived subfraction that show chemotherapeutic properties affecting the viability of G cells and healthy PBMCs. And we have used NMR to identify which compound was responsible of the activity, identifying the pyrogallol-bearing compound as the most active compound. The next steps are, of course, to further the further study of pyrrologallolpyrin compound in yeast to evaluate their additive and synergistic effect, deciphering the mechanism of action of this compound in yeast, and of course evaluating efficacy and toxicity in animal models. To conclude, with acknowledgement, I want to thank my group in Bologna, Professor Elia, and my group here in Napoli, and of course all of you for your kind attention.